In Chapter 9, we're going to cover cellular respiration and fermentation. The chapter really is going to have a shift in the way that we've been doing the biology. Instead of memorizing a random list or trying to memorize all these facts, what we're going to do is we're going to tell a story. So we're going to start at one point, and then we're going to follow that process along. Cellular respiration is going to be in the majority of the chapter. About 3/4 of the chapter is cellular respiration, and then the last quarter we'll look at a related process, which is fermentation. So just to give ourselves a big picture view of what we're trying to do in chapters 9 and 10 is we're thinking about how cells use energy. So all living cells require energy, and they do this in multiple ways. Some organisms use photosynthesis, and all eukaryotic organisms have the ability to do cellular respiration. Looking over this chart that we have on the slide, you can see that what we do is we take in organic molecules and oxygen, and then we use our mitochondria to transform the energy that's in that organic molecules into ATP, which can be used, and then we're left with carbon dioxide and water as a endpoint. This process is what we mean when we say cellular respiration. So that's the part of the process that we're learning about today, this bottom half of the circle, where we take organic molecules in the presence of oxygen, completely break them apart, release lots of energy in the form of ATP, and then we're left with carbon dioxide and water. Related to this process, though, is what plants can do, which they will take the carbon dioxide and water that's in the atmosphere, they process that in the chloroplast, and they're able to bring light energy in. And what plants are able to do that's really impressive is they're able to take that light energy and they're able to capture it in organic molecules. And so you can see the end products of one become the starting products for the other. And they're very interconnected. Even though we talk about them in chapter 9 and then chapter 10, these processes are a way for organisms to use energy. So we want to remind ourselves some things before we start looking at cellular respiration. Cellular respiration is a catabolic pathway, meaning it's going to release energy. It's going to break down. It's exergonic. If we think about our little reaction, it is this one, right? It starts off with big, high energy quality starting molecules, and then we release the energy over time. Delta G is going to be negative. It's spontaneous. As we think about how this catabolic pathway works, it works because of electron transfer. We're going to move electrons around, and as we move those electrons around, that's going to release energy. There are 3 different catabolic pathways. So 3 different ways that we can take food and we can break it apart to release energy. The first one that we're going to talk about is called aerobic respiration. Aerobic respiration is when you take your organic molecule, your food, and you completely break it down in the presence of oxygen, and this generates a lot of ATP. When I talk about cellular respiration throughout this chapter, I am specifically talking about aerobic cellular respiration. There's another process called fermentation. We'll talk about it a little bit in this chapter. Fermentation is where you take an organic molecule, you break it apart just a little bit, and you do this without the presence of oxygen. It's going to provide a little bit of ATP, but not lots. There is this 4th thing that I want to mention exists, but we are not going to talk about it in greater detail. There is something called anaerobic respiration. This is a process whereby organisms are able to take organic molecules, break them completely apart, release lots of oxygen, but there is no oxygen present. So I want you to know that such a thing exists, but we are not going to look at that pathway in any more detail in this particular class. So for us, really the big points that we want to focus in are these first 2, the aerobic respiration and the fermentation. Let's begin by talking about cellular respiration. Again, that aerobic cellular respiration. Listed on your screen is the overall chemical equation for cellular respiration. You need to memorize this. You must know this overall chemical reaction. So we are going to pretend that every organism just eats very simple molecules. So we start off with an organic molecule of glucose, C6H12O6, that's the food, plus oxygen. So those are the 2 things that go in. You need to eat and you need to breathe. Your body does cellular respiration, and then we get 3 outputs. Those 3 outputs include carbon dioxide, right? What we breathe out. Water, imagine needing to go to the bathroom really bad. And then more importantly, we make energy in the form of ATP and heat. We're going to make about 32 ATP molecules for each one glucose molecule. Heat is also released, but just as a reminder, heat is that very low quality energy that it's harder for us to use. So again, I really encourage you to memorize this equation. This is a good big picture view of what we're doing with aerobic cellular respiration. So again, 2 things in, glucose and oxygen, and then 3 things out, carbon dioxide, water, energy, and heat, and then energy in the form of ATP and heat. To set up our discussion of cellular respiration, we need to talk about redox reactions. As we work through a aerobic cellular respiration, we're going to see the transfer of electrons. As those electrons are moved around, this is going to release energy. So when you are moving electrons around, these are called oxidation and reduction reactions. Oxidation and reduction. Chemists have gotten tired of saying oxidation, reduction over and over and over again, and so they've taken these 2 words and sort of smooshed them together, and what they talk about are these things called redox reaction. You can hear reduction, oxidation happening. If there is an oxidation, there's always a reduction somewhere. Likewise, if there's a reduction, you have to have an oxidation always also happening. Oxidation means that you are losing electrons. Reduction means that you are gaining electrons. Note that it's one of those cases where science isn't really quite making sense, right? Reduction is gaining electrons, so that's a little bit odd. An easy way to remember redox reactions is with the mnemonic, Leo the lion says grr. So you got to spell it this way or it doesn't work. The reason this is going to be useful is if we think about Leo, if you lose electrons, you're oxidized, so that's Leo. And then grr, if you gain electrons, you're reduced. Down here, you can see a nice equation of this. We've got X, what's happening? It's lost the electrons, so it's been oxidized. Whereas on the other hand, Y has gained the electrons, so it's being reduced. And we're going to see this happen in cellular respiration. Cellular respiration is a redox reaction. Things are going to be losing electrons and other items are going to be gaining those electrons. And this is important because each time those electrons are moved around, that's releasing energy that we can capture to do work. Unfortunately, not all cellular reactions or equations show you easily where the electrons are. So one good way to know where the electrons are moving is to follow the hydrogens. If something has lots of hydrogens in it, you can assume that it has lots of electrons. If it has less hydrogens in it, it has less electrons. So looking at this equation down here, we've got methane with lots of hydrogens, so lots of electrons, and it's becoming carbon dioxide. So in this case, it's losing the hydrogens or it's losing the electrons, so we say that it is oxidized. On the other hand, oxygen, as we look at it, it's starting off with not any hydrogens, and now it's turning into water where it's gaining those hydrogens. So if it's gaining hydrogens, it's like gaining electrons. So oxygen is becoming reduced. Aerobic cellular respiration is a redox reaction. So let's see if you can use these terms the right way. Glucose is going to become carbon dioxide. Right now, just go ahead and mutter it to yourself or say it in your mind. What is happening to glucose? Is it being oxidized or is it being reduced? Hopefully you saw that glucose had lots of hydrogens and then it was losing those hydrogens. If you lose hydrogens, that's called being oxidized. Let's try the other one. If there's an oxidation, there has to be the opposite reaction happening. Here we can see oxygen gaining those oxygen turning into water or gaining those hydrogen ions. Here's our reduction reaction. So again, cellular respiration is a redox reaction. Oxidation means you've lost the electrons. Reduction means you've gained the electrons. Here we see our overall cellular respiration equation again that is a redox reaction, and you can see that glucose is being oxidized and oxygen is being reduced. It turns out that organic molecules like glucose, things that have lots of hydrogen in them, have lots of electrons that can be transferred to oxygen. Unfortunately, this process has to happen in a stepwise fashion. So let's just imagine if you have a molecule of glucose and you try to release all of its energy in one big explosion, it can release over 686 kilocalories per mole of glucose, which I know probably means nothing to you, but that's a really big number. ATP is only 7, right? So one piece of glucose has tons of energy in it. You can think of it if you've ever accidentally burnt something that contains sugar on your stove, right? Think about that. It'll explode in this big explosion of light and heat. Unfortunately, we don't need an explosion of light and heat when we release the energy from glucose. What we want is we want to take the glucose and we want to release, poof, a little bit of energy. Poof, a little bit of energy. We don't want explosion of light and heat in our bodies. So what we're going to have to do is somehow we're going to have to take this glucose molecule and we're going to have to release its energy in some sort of stepwise fashion. What we'll do in cellular respiration is we'll take glucose and we will break down the molecule in a series of steps. Instead of releasing all of the electrons at once, we're then instead going to poof, release a little bit of energy. Poof, release a little bit of energy. What we often do with the electrons in glucose and or other organic compounds is we usually transfer them to something called NAD+. This is a coenzyme. This coenzyme has two different forms. The way it's often drawn and the way that we'll talk about it in class is think of it as a box. Okay, so this box down here is NAD. NAD comes in two different forms. One version is called NAD+. NAD+, think of this as the empty box. It doesn't have any electrons. There's another form of the box called NADH that is similar, but what it has is it also contains electrons. So what we have is this molecule now that can be oxidized or reduced. Again, if you've lost your electrons, you're oxidized. If you've gained your electrons, you're reduced. And so what happens in cellular respiration is we're going to start with glucose. We're going to take some electrons away from glucose, and then we're going to put those electrons into this box, into NAD+, to turn it into NADH. These boxes of NADH will then be transferred to something called the electron transport chain, where those electrons will continue to be allowed to release energy. So now I want to draw out the electron transport chain, so you'll get to stare at my ceiling while I lay this down so I can draw a little bit better. The electron transport chain is a series of proteins embedded in the mitochondrial membrane. For simplicity's sake, we often draw the electron transport chain like a staircase, like you can see on this particular slide. What happens in the electron transport chain is we're going to bring in those molecules of NADH that we've already had. So those are the boxes, right? So we bring in a box of NADH that has the electrons on it. And what happens is NADH will drop the electrons off onto the electron transport chain. Once NADH has dropped off its electrons, then the oxidized NAD+ will float off to get filled back up. But really what I want to focus on is what's happening with those electrons. So NADH comes in, it drops off the electrons onto the electron transport chain. The electrons are going to sort of fall down the electron transport chain, much like a ball falling down a set of stairs. So this electron will fall down, ploop. As that electron falls down, this is going to give off a little boost of energy, poof. And then it's going to do the exact same thing. That electron will fall again, and poof, little burst of energy. That electron falls again, poof, little bits of energy. The reason that the electrons are falling is not because of gravity, it's because of what's happening at the bottom of the electron transport chain. At the bottom of the electron transport chain, we have a gigantic molecule of oxygen. Oxygen is a very electronegative, meaning that it's hogging the electrons towards itself. So the oxygen down here is drawing all of the electrons towards it. And so that electron will eventually get down to oxygen. When the electron gets down to the oxygen, the electron plus a hydrogen plus the oxygen will form some water. And so in this process now, we've got our electron transport chain where we've taken those electrons that were in the NADH molecule, we've put those electrons onto the electron transport chain. As those electrons fall down the electron transport chain, they're giving off poof, 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 little amounts of energy. And the reason that those electrons are falling down is because of this molecule of oxygen that's at the bottom that's drawing those electrons towards itself. This is really important because what it's done is it's allowed us to release the energy in small, manageable amounts instead of one explosive release. So in review, our electron transport chain is passing the electrons down in a series of steps. The reason that the electrons are moving through that electron transport chain is because oxygen is pulling the electrons down. Those little poofs of energy are going to be used to generate ATP. So if we think about the path that our electrons are taking, first the electrons were in our food, then we put them into an NADH box, then they were put onto the electron transport chain, where eventually they combine with oxygen to form water. You should know that that's sort of the path that these electrons are taking through the process. Now that we've set up some of the background to understand cellular respiration, we want to look at cellular respiration in further detail. We can take cellular respiration and divide it into 3 stages. The 3 stages of cellular respiration include glycolysis, which will always be colored with this background of teal in your notes. It's really nice. A second step called pyruvate oxidation in the citric acid cycle. Yes, it's 2 things, but we talk about those at the same time. So that's the middle step, pyruvate oxidation and the citric acid cycle. That's step 2. And then our third step is oxidative phosphorylation. With these stages of cellular respiration, you absolutely need to know these stages, where they occur, and how we make the ATP in each step. So 2 really important things already that I've asked you to know well for the exam in this class. First, you should know the overall chemical equation for cellular respiration. And then secondly, you should know the stages of cellular respiration, their locations, and how the ATP is made. So here we can see a nice table that we would normally do out on the board to fill out, but here it is on a nice slide where we can see the 3 stages of cellular respiration, their location, and how the ATP is made. We'll go through each. The first stage, glycolysis, takes place in the cytosol. This is important to point out that this does not require any specialized organelle. It doesn't need a mitochondria. Every cell has cytosol, and so every cell is at least able to do glycolysis. We are going to generate a little bit of ATP in this process of glycolysis. We make that ATP through a process called substrate level phosphorylation, which we'll talk about in a moment. The second step, pyruvate oxidation in the citric acid cycle, does require a specialized location. This is going to take place in the innermost part of the mitochondria or the matrix of the mitochondria. So step 2 takes place in the matrix of the mitochondria. This also generates a little bit of ATP through that same process of substrate level phosphorylation. The third and final step is called oxidative phosphorylation. Oxidative phosphorylation takes place across those folds of the inner membrane of the mitochondria, or across the cristae of the mitochondria. In oxidative phosphorylation, we actually generate tons of ATP, and we do it through a unique process called oxidative phosphorylation, and we'll learn what that means as we talk through this third step. Again, this is a really important chart that you should understand. You should know the 3 stages of cellular respiration, where each stage occurs, and how the ATP is made in each step. Those of you guys that like a visual representation of it, here is the vision from your textbook. So over here we've got glycolysis happening. Glycolysis happens out in the cytosol, and we make a little bit of ATP through the process of substrate level phosphorylation. The next step is pyruvate oxidation in the citric acid cycle. Pyruvate oxidation and the citric acid cycle take place inside the matrix of the mitochondria. So we can see step 2 happening there in the matrix of the mitochondria. And this is also going to make ATP, but this is going to make ATP through substrate level phosphorylation as well. The third and final stage is oxidative phosphorylation. Oxidative phosphorylation takes place across these folds of the inner membrane, across the cristae. This is actually going to generate lots of ATP. You can see a really, really large ATP explosion there, because what we're doing is we are using this process called oxidative phosphorylation. So there's 2 different ways, as you saw on that list, to generate ATP. The first way is through substrate level phosphorylation. The word substrate hopefully sounds familiar. It sounds like that hand fist demonstration that we did in class. In substrate level phosphorylation, there is an enzyme that is synthesizing the ATP. You can see that down here at the bottom of the screen. So we've got our enzyme, and what does it do? It takes a phosphate group and sticks it onto an ADP molecule, and ta-ta, we make ATP. So there's just an enzyme that's sticking molecules together to make ATP. That's how we make the ATP in both glycolysis, the first step, and the citric acid cycle pyruvate oxidation, second step. The third step is oxidative phosphorylation. Again, we'll talk about that when we get to the details of the third step. This is actually, though, where we're going to make most of our ATP for aerobic cellular respiration. You should also know this relationship: 1 molecule of glucose into cellular respiration equals about 32 molecules of ATP out. So what I want to do now is I want to just take you through each of these steps and sort of explain what's going on. We'll start with glycolysis. Glycolysis is going to break down glucose into 2 molecules of pyruvate. Glycolysis occurs in the cytoplasm whether or not oxygen is present. And as we look at glycolysis, we'll want to be really careful about understanding the difference between net versus total ATP production. In the space, I'm going to try to diagram out glycolysis for you the similar way that we would on the board. Some things to remember about glycolysis is that glycolysis happens in the cytoplasm and that it is making the ATP through substrate level phosphorylation. In glycolysis, we're going to need to start off with a glucose molecule. So we need to start off with glucose. In order to draw a glucose molecule, what we're going to do is we're going to draw 6 circles. 1, 2, 3, 4, 5, 6. That represents the 6 carbons that are in that glucose molecule. In the process of glycolysis, glycolysis, sugar cut, we're going to take this glucose molecule and we're going to chop it in half. And so what we end up with are 2 3-carbon molecules. Let me make that a little bit more obvious there. So in glycolysis, we take that glucose molecule and we break it up into 2 3-carbon molecules. Those 2 molecules that we broke the glucose into are called pyruvates. So in glycolysis, we start off with a glucose and we break it into 2 pyruvates. Just like any reaction, this is not going to happen magically. There is an energy investment. So in order to get this spontaneous reaction, we have to put in some oomph, right? We have to put in some energy to get it started. The energy that's required for this process to get started is 2 molecules of ATP. In the process of taking glucose and breaking it apart into 2 pyruvate molecules, we do generate some ATP. We generate 4 ATP. And we also make some of those boxes that are holding the electrons. We're also going to generate 2 NADH molecules. This process is actually going to have lots of different intermediaries with lots of different enzymes. I'm not interested that you understand all of those intermediaries or enzymes except for one enzyme. There is one enzyme that works right here in the middle that's going to be important to know. And this guy has a long name, but it's the only one you have to know. So this enzyme that works here in the center that is one of the enzymes that helps us break the glucose into pyruvates. That enzyme is called phosphofructokinase. It's a nice long word there. So if we walk our way through this process, let's just start at the beginning. We're talking about glycolysis. It happens in the cytosol. We're making some ATP through the process of substrate level phosphorylation. We begin with a glucose molecule that's 6 carbons big. It gets broken into 2 3-carbon molecules of pyruvate. The energy investment is 2 ATP in. We get 4 ATP and 2 NADH out. And one of the enzymes that helps this process is an enzyme called phosphofructokinase. You need to be really careful when we talk about the energy that is produced in glycolysis, when we talk about the difference between net and total ATP produced. So you can see that we put 2 ATP in, but we got 4 ATP out. So really what we walked away with was just 2 ATP. So our net ATP production was 2, whereas our total ATP production was 4. Do watch out for that when you especially are working in the homework. Pearson likes to be very careful about the way they word the questions, but if we ask you about glycolysis, always check to see if they're asking you about the ATP that's produced, if it's the net ATP produced or the total ATP produced. On this page, you can see a similar diagram that your textbook uses to describe glycolysis. And you might notice that there's some more information on this slide than what was on the previous picture we drew. You do not need to understand any of the additional information that's in this particular slide. Please use the diagram that we drew to let you know which are the most important molecules to look at. But to just help you through the slide, let's go through it. We start off with a glucose molecule. You put 2 ATP in, you get 4 ATP out and 2 NADH, and you end up with 2 pyruvates. I will never ask you about what happened to these electrons or what happened to this hydrogen ion. We really just want to focus on that main part of the molecule process that we diagrammed out before. Do remember that we have that one little enzyme though that's really important to remember here that works in the middle. That enzyme's name is phosphofructokinase. Your textbook really gets overexcited about cellular respiration, and it shows you the entire biochemical process of glycolysis with this scary picture that's coming up next. You do not need to understand all of this detail. I am never going to ask you about glucose 6-phosphate or phosphoglyceromutase. If you were ever asked questions about these, I would assume that you would just Google it like every other biologist. However, there is one really important element. You do need to know that phosphofructokinase is one of the enzymes that helps us take glucose and break it up into the 2 pyruvates that we end up with at the end. Now that we're done with the first step of glycolysis, we want to move to the second step. So remember, the second step is pyruvate oxidation in the citric acid cycle. These are happening in the matrix of the mitochondria. These are going to require oxygen, and this is where we're taking those pyruvate molecules and we're changing them to acetyl-CoAs. Again, I'd like to try to draw this out for you. In pyruvate oxidation, what we want to do is we want to take the pyruvates from glycolysis and get them into the mitochondria. So up here in black, you can see glucose getting broken up into those 2 molecules of pyruvate. That was glycolysis happening out in the cytoplasm. And then I've drawn a really zoomed in picture of a mitochondria. So that's what's in red down here. What we need to do is we need to take these pyruvate molecules that are out here in the cytosol, and we need to move those pyruvate molecules into the interior of the mitochondria, into the matrix. And that's going to be called pyruvate oxidation. As we do this, we are going to bring the pyruvate molecules into the matrix of the mitochondria. They are going to get changed a little bit. Instead of still being 3 carbons big, they're going to each lose a carbon. So they're going to be 1, 2 circles big, 1, 2 circles big. So in the process of bringing the pyruvates into the mitochondria, we lose 2 carbon dioxide molecules because 2 of those carbons were left. As those pyruvates come into the mitochondria and they lose those carbon dioxides, they also bind to a coenzyme. The coenzyme that they bind to is called coenzyme A. Coenzyme A. In this process of bringing the pyruvates into the mitochondrial matrix, it does not generate any ATP. However, some electrons are released and they get captured. So in this process, we also generate 2 molecules of NADH. These little 2 circle molecules down here with the CoA on them have a name. I just gave it away. These are called acetyl-CoA molecules. And that process, as we've drawn it out, is pyruvate oxidation. So just walk through it again. We start off with our 2 pyruvate molecules. We're going to bring them into the mitochondrial matrix, and they are going to change into 2 acetyl-CoAs. And the process of taking the pyruvates from the cytosol and bringing them into the mitochondrial matrix, this is going to generate 2 carbon dioxides and 2 NADH molecules. Here's the picture of pyruvate oxidation from your textbook. It sort of flipped our images on its side. Here we start off with a pyruvate. As it enters the mitochondria, it gets changed into an acetyl-CoA. In the process of doing this, it generates a carbon dioxide and an NADH You'll notice on this picture, it's only showing one pyruvate getting changed into one acetyl-CoA, so it only shows one carbon dioxide and one NADH getting produced. In reality, though, these numbers would need to be doubled because we do have two pyruvate molecules coming into the cell. After we've done pyruvate oxidation and brought that pyruvate into the mitochondria and changed it into an acetyl-CoA, we now need to go through the citric acid cycle. The citric acid cycle is sometimes also called the Krebs cycle for Mr. Krebs. He was the scientist who discovered its mechanisms. The citric acid cycle is going to require oxygen, and it's the citric acid cycle that's actually going to make some ATP. Remember, in the pyruvate oxidation, we really only made carbon dioxide and some of those NADH molecules. In the citric acid cycle, we're still working inside the mitochondria. So you can see my big red mitochondria. From the previous step, we brought in our two acetyl-CoAs. And in the citric acid cycle, it's a cycle, meaning it's going to be a circle. We're going to start and end with the same product every time we go around it. The name of the molecule that we are going to start with and end with every single time is a 4-carbon molecule called oxaloacetate. Yes, you need to know that word. What we're going to do is one acetyl-CoA is going to come into the citric acid cycle, and it's going to combine with one oxaloacetate. So you're bringing in one acetyl-CoA, and it's combining with one oxaloacetate. If we just count up the carbons, we've got 1, 2, 3, 4, 5, 6. So what we create is this 6-carbon molecule. This 6-carbon molecule that we have created is a molecule called citrate. That's the reason for the name of this cycle being called the citric acid cycle. We're then going to take citrate, and through a series of reactions, we're going to completely pull it apart. We're going to rip out every single little electron that we can. And as we do that, we're going to generate 4 byproducts. You should know those 4 byproducts. We are going to generate the following. We are going to make 3 molecules of NADH. We will generate 1 molecule of ATP, 1 molecule of FADH2, and 2 molecules of carbon dioxide. So we've talked about NADH molecules, that top molecule already. Those are the boxes that are holding the electrons. We generate just a little teeny bit of ATP, and then we make this new thing, FADH2. FADH2 is another electron carrier. It's just a little bit lower quality than NADH2, but just think of it as a box. FADH2 is just this other thing that is holding electrons for us. We also generate 2 carbon dioxides. If we look back, citrate was 1, 2, 3, 4, 5, 6 carbon molecules big. If you take citrate and move it around and then remove 2 carbons, 6 minus 2 equals 4, you're right back to oxaloacetate, and the process repeats again. In order to make the citric acid cycle work for a glucose molecule, you're actually going to have to do the citric acid cycle twice. You're going to have to do it 1 time for the first acetyl-CoA, and then you'll have to do it a 2nd time for the 2nd acetyl-CoA. The numbers that I gave you over here, those are the numbers that show what happens in 1 turn of the citric acid cycle. We would need to double those numbers if we were talking about the output that would come from a complete glucose molecule. Here's the complete picture of pyruvate oxidation and the citric acid cycle. So up here we've got the pyruvate molecule being broken into acetyl-CoAs. That's generating carbon dioxide and NADH. From here we can see the citric acid cycle. Let's start with oxaloacetate. Oxaloacetate comes in. It's going to combine with 1 acetyl coenzyme, A, to make a citrate molecule. We're then going to break that citrate molecule apart. As we break that citric acid molecule apart, 4 items are going to be generated. We're going to generate 2 carbon dioxides, 1 ATP, 3 NADH, and 1 FADH2 molecules per turn. This process will need to happen twice per 1 original glucose molecule. Once again, your textbook gets a little overexcited about the citric acid cycle, and it shows you in a little bit too much detail the process. process. So here's the scary diagram of the citric acid cycle. Again, you do not need to know this level of detail. If for some reason in Pearson, especially as you're working through the dynamic study modules, any of these weird intermediary molecules come up in a question, please just Google the answer. I would never expect you to know these intermediaries. I would just expect you to use the internet if you were ever asked a question about them at this point. This brings us to our third step, oxidative phosphorylation. Before we start talking about oxidative phosphorylation, we just want to think about what we've done so far. So we're doing aerobic cellular respiration. We know that one glucose molecule in is going to give us 32 molecules out. However, if we look at what we've done in glycolysis, and then that second step, the pyruvate oxidation and the citric acid cycle, we really haven't made very many ATP molecules. We're supposed to be making 32. We've got just a small number. What we've really done in the first two steps is we have made lots of molecules of FADH2 and NADH. And so what we need to do in this third and final step is we need to move these electrons to our electron transport chain. We already sort of saw the electron transport chain earlier, and what I want to do is revisit that and show you how it works in this third step. So the electron transport chain is located in the inner membrane or the cristae of the mitochondria. If we think about a mitochondria, right, here's my mitochondria, and there's those cristae. Where this electron transport chain is it's going to be located in the folds of that inner membrane. So that staircase is in the inner membrane or in the cristae of the mitochondria. When we talk about the electron transport chain and we keep drawing the little zigzag, it's not an actual zigzag. These are actually proteins in the mitochondria that can alternate reduced and oxidized. Leo the lion says grr. So what the proteins can do is they can gain an electron. Imagine that my pen is an electron, right? I've gained the electron, I'm reduced, and then I've lost my electron. Oh, I'm oxidized. Oh, but then I gained an electron, I'm reduced. Oh, but I lost it. And this is what's going to keep going on over and over. You're reduced, oxidized. This process absolutely requires oxygen because oxygen is that final electron receptor of this electron transport chain. All right, let's finally learn about this third step, oxidative phosphorylation. Again, this takes place across the inner membrane of the mitochondria, and this is going to generate lots of ATP for us in a process called oxidative phosphorylation. Let's understand sort of what I've got drawn out here, and then we'll add some elements to it. Oxidative phosphorylation is this entire picture. However, oxidative phosphorylation can be divided up into two parts. There's the first part called the electron transport chain, and then there's a second part called chemiosmosis. And so oxidative phosphorylation is actually both components. We're going to first transport the electrons, and then second, we're going to do this thing called chemiosmosis. To help you out with the orientation a little bit, the black line that's running through the middle, you can imagine this as being the inner membrane of the mitochondria, sort of like what we drew on the previous slide. Down here at the bottom of the slide, this is where we're in the matrix. Up here at the top of the slide, this is where we're in the intermembrane space. So what happens in oxidative phosphorylation is we're going to begin with the electron transport chain. NADH and FADH2, those electron carriers, will bring in their electrons and drop their electrons off on the electron transport chain. You can notice that, that FADH2 is not as high quality of a molecule, so it's dropping off its electrons at a slightly lower position on the electron transport chain. As these electrons are dropped on the electron transport chain, they will then begin to fall down the electron transport chain. So as they fall, poof, they give off some energy. The electron falls and poof, it gives off some energy, and those electrons continue to fall all the way until they get to the bottom of this. When those electrons combine with oxygen, they'll be added to some protons or some hydrogen ions to form water. That process right there, that's the electron transport chain. So let's walk just through the electron transport chain moments again. We begin up at the top with NADH and FADH2 dropping off electrons on the electron transport chain. As they drop off their electrons, the electrons will then fall down the electron transport chain. They fall because oxygen is our final electron acceptor down here at the bottom. When the electrons combine with the oxygen and hydrogen, this will form a water molecule. In this process, what we're doing is we're giving off little bursts of energy. Note that still, as we've talked about this electron transport chain, we have not made any ATP yet. All we've done with the electron transport chain is gone poof, 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 and given off those little puffs of energy. Now let's talk about the chemiosmosis part. Here's where we're finally going to make some ATP, and we'll talk about what we're doing with those puffs of energy. With these little puffs of energy that we're making, poof, 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 what we're going to do is we are going to actively pump hydrogen ions that are in the matrix across the matrix and into the intermembrane space. And so with every little poof of energy, we're going to pump a hydrogen across the membrane. Hydrogen, hydrogen. And we are just going to continue pumping hydrogens across. Sorry, as you can't tell, those are H's anymore. We are going to keep pumping hydrogen ions across the membrane, and we're going to build up this gigantic concentration gradient of hydrogen ions in the intermembrane space. So if we look at it now, I've got a lot of hydrogen ions now that are in the intermembrane space. I have very few hydrogen ions that are down in the matrix. And so what we learned in chapter 7 about diffusion is that these hydrogen ions are going to want to diffuse back across this membrane to the matrix. As they do this, as the hydrogen ions automatically diffuse back across the membrane to get into the matrix, they are going to flow through ATP synthase. ATP synthase is an enzyme that looks like a spinny rotor. We'll look at a better picture of it in a moment. And what happens is it spins around sort of like a rotor on a boat. And as it spins around, this is going to generate tons and tons of ATP. So it's this entire process of the electron transport chain, moving those electrons down, releasing those poofs of energy, and chemiosmosis, using those puffs of energy to pump the hydrogen ions from the matrix into the intermembrane space that creates the ATP. So it's this entire picture is what we mean when we talk about oxidative phosphorylation. It would be really important to know the locations of these molecules. Please again remember that the hydrogen ion concentration is low in the matrix and it's high in the intermembrane space. Or another way of saying this might be to say that it's going to be acidic out here in the intermembrane space because there's lots of hydrogen ions, and it's going to be more basic out here in the matrix because there's a lower hydrogen ion concentration. So we've seen our our oxidative phosphorylation. That entire process that we were just drawing out, just to remind ourselves, is happening across the inner membrane of the mitochondria. So that zigzag staircase is happening in that inner membrane where we are going to have a low amount of hydrogen ion concentration in the matrix, and then we are going to have a high hydrogen ion concentration out here in the inner membrane space. Again, not the greatest picture, but hopefully that helps you sort of orient the previous picture. And again, the thing to remember about oxidative phosphorylation is that we are making tons of ATP in this third and final step. Here's what the electron transport chain actually looks like. So these are the staircase that we were drawing out before. What happens is that the electrons are releasing free energy as they pass from protein molecule to protein molecule. The reason that they're moving is because there is a molecule of oxygen down here at the bottom that's drawing those electrons through this protein carrier chain. Once they bind with oxygen, they'll then form water. The electron transport chain is not making any ATP. What the electron transport chain is doing is it's releasing energy in those poofs, those small, manageable amounts. Chemiosmosis is where we're going to take the energy released from the electron transport chain and pump those hydrogen ions or those protons from the matrix into the intermembrane space. In this picture, the protons or the hydrogen ions are those gray balls. What happens is once you've built up a hydrogen ion concentration in the intermembrane space, those hydrogen ions then flow through ATP synthase. That's this purple molecule that you see drawn right here. As that molecule spins, as those hydrogen ions are flowing through it, this is going to generate ATP, and this process is called chemiosmosis. If you're a real visual learner, there is a great animation in Pearson in Mastering Biology where you can watch this entire process. It's called a bioflix. I really encourage you, especially if you know you're a real visual learner, to watch this process. I think it'll help tie the entire process together for you. So we're now done talking about aerobic cellular respiration. We can think about how the energy has moved through the process. First, the energy was in the glucose. Then we put it into those boxes. That energy went down the electron transport chain. We used that energy, those poofs, to create the proton motive force that's moving the protons across the membrane. And then we put the energy in ATP. This process is about 34% efficient, which might at first sound really wasteful, but this is actually really efficient. Even the best car engines are somewhere usually in the 20s of their efficiency. So this process actually is able to capture quite a lot of the energy that's in our food molecules and convert it into ATP. Notice that I keep saying the following. I say 1 glucose molecule in gives you about 32 ATP out. There's a reason that this is approximation. I'll give you the 2 reasons right now. Number 1, sometimes when your body is doing this process, you are going to use some of these molecules for other processes in the cell other than just making ATP. So you might be in the middle of breaking apart glucose into pyruvates, and your body might say, pyruvates, I could use those to go build some bone. And so obviously we won't make so much ATP if we're going to use those pyruvates for some other process. The second thing that happens has to do with these 2 NADHs that are created in the process of glycolysis. Those NADH molecules are out in the cytosol. They have to get all the way into the mitochondria to do oxidative phosphorylation. Depending on how these are moved in the mitochondria, sometimes we're going to lose or gain more energy. So the rule of thumb is 1 glucose molecule gives you about 32 ATP. It's not always a precise number, but that's our best estimate. Now we want to discuss what's going to happen when there's no oxygen present. Remember, there is that weird thing called anaerobic respiration. We're not going to talk about it at all. For us, when there's no oxygen present, we want to understand the process of fermentation. Fermentation will begin with substrate level phosphorylation to generate some ATP. That's a really fancy way of just saying glycolysis. It's not as scary as it sounds. And then we have to have a step where we regenerate the NAD+ molecule. There are 2 types of fermentation that I want you to become familiar with. The first one is called alcohol fermentation. The second one is called lactic acid fermentation. Alcohol or ethanol fermentation is going to begin with glycolysis. So the first part of this process should look really familiar. We begin with a molecule of glucose. There's our 6-carbon glucose up at the top. And we break it up into 2 molecules of pyruvate down here at the bottom. In this process, we generate 2 net ATP. Notice I'm showing you just the net ATP. And we generate 2 NADH. All cells are able to do this process. It doesn't require a mitochondria, and all cells are able to do this because it doesn't require the presence of oxygen. So for the cell that's living in some sort of environment where there's no oxygen, this is great because what the cell has done is now it's generated at least 2 ATP from its food instead of dying. This part, though, we have a problem. We've generated these 2 NADH molecules, those boxes holding electrons. But there's no electron transport chain because there's no oxygen. And so what's going to happen is we're going to begin to accumulate all these boxes full of electrons, and that's not sustainable. So I have to do something with those NADH molecules. And here's the added little step. What we're going to do is we're going to take the electrons from NADH and we're going to dump them out on the pyruvates, and we're going to regenerate just regular old NAD+ molecules. We'll have 2 of them. So I've taken the electrons from the NADH and I've dumped them out on the pyruvates to give me 2 empty boxes in the corner. Sorry, that's really hard to read. I'm sorry, my handwriting is so bad here on the tablet. So we've got NADH. well, we don't have anything. We have NAD+ molecules. In the process of dumping the electrons out on the pyruvates and regenerating these two NAD+ molecules, this changes the pyruvates. The pyruvates get changed into two carbon molecules. Those two carbon molecules are called ethanol. In the process, you can see that we've lost some lost some carbons. So we started off with 1, 2, 3 carbons. Now we only have 1, 2. We started off with 1, 2, 3. Now we only have 1, 2. So we've lost 2 carbons. So also in this process, it generates 2 carbon dioxides. That right there is ethanol fermentation. I know it got a little bit sloppy, so let's make sure we really highlight the endpoints that we want to know. Alcohol fermentation is taking glucose and breaking it apart through glycolysis to generate 2 ATP. That's the exciting part for the organism. This is how it's not going to die when there isn't oxygen present. In the same process, though, it has to then take care of those NADH molecules. So it is also going to generate ethanol, carbon dioxide, and it will regenerate 2 NAD+ molecules. Here we can see the picture that your textbook uses to describe alcohol fermentation. Again, we start off with glucose molecule. That glucose molecule is getting changed into 2 pyruvates, and that process will generate 2 ATP. From there, we have to take the NADH molecules that were created and regenerate them. As we do this, this will create 2 ethanols and 2 carbon dioxides. Really, as we think about alcohol fermentation, what we want to think about is these 4 products. In alcohol fermentation, we're going to generate alcohol, CO2, ATP, and NAD+. Next, we have lactic acid fermentation. It's going to start off the exact same way. This should look really familiar. We start off with a glucose molecule that's broken into 2 pyruvates. This generates 2 net ATP and 2 NADH. Again, for the cell, it's really excited. It made some ATP even though there wasn't oxygen present, and it's not going to die. The problem is the same as before. We've got NADH molecules that are full of electrons, and we need to do something with them since there's no oxygen and we can't use an electron transport chain. Once again, we're going to take those NADH molecules and dump those electrons out on the pyruvates and regenerate the NADH molecules and create 2 NAD+ molecules. In the process of dumping the electrons out on the pyruvates, we're going to do something slightly different with the lactic acid fermentation. The pyruvates are going to change, but watch how it looks different this time. The pyruvates are going to get changed into another 3 carbon molecule. We're just changing the arrangement of those carbons. So we haven't lost any carbon in this process. These 3 carbon molecules that we created down here are called lactate. And so this is the reason that this process is called lactic acid fermentation. So again, big picture, what have we made? We've made ATP for the cell. We've also generated 2 molecules of lactate, and we regenerated 2 NAD+ molecules. Here's the image from your textbook of the same process, lactic acid fermentation. First, we do glycolysis. We take glucose and turn it into 2 pyruvates. That generates 2 NAD+ and 2 ATP. In that process, the cell generates the energy. Unfortunately, it then has those NADH molecules that have to be regenerated. So we're going to take the electrons from NADH, dump them out on the pyruvates, and that leaves us with 2 lactate. So if we think about the lactic acid fermentation process, we generate 2 ATP, 2 lactate molecules, and 2 NAD+ molecules. From here, there is a Science Friday video to watch. I'm going to make a link in Canvas with all the Science Friday videos for this particular unit. I encourage you to go enjoy Sandor Katz and his love of fermentation. At this point, we can now compare and contrast what we did in the first 3/4 of the lecture with this last portion. Aerobic respiration and fermentation do have some things in common. They both start off with glycolysis. That's exactly the same in both processes. Another thing they have in common is that NAD+ is their electron acceptor. They're using that box, NAD+, that coenzyme, to accept electrons. However, there are some big differences. In fermentation, The final electron acceptor was an organic molecule, something like the lactate or the ethanol. That wasn't the case in aerobic cellular respiration. In aerobic cellular respiration, your final electron acceptor was oxygen. So that's a difference. Another big difference is the amount of ATP that's produced. Fermentation only produces 2 ATP per glucose molecule. Cellular respiration, on the other hand, can produce 32 ATP. So if organisms have the choice to choose between doing fermentation and aerobic cellular respiration, aerobic respiration is really the one that they're going to prefer to do because they get so much more bang for their buck, or they get so much more ATP per each glucose molecule. We classify organisms on what type of pathway they're able to perform. So obligate aerobes, you're obligated to be aerobic. Aerobic means with oxygen, are those organisms that have to use cellular respiration or else they'll die. You and I were obligate aerobes, right? We have to do cellular respiration. There are also organisms called obligate anaerobes. These are organisms that are poisoned by oxygen. So these are organisms that always do fermentation, or that weird third thing that we're never talking about called anaerobic respiration. And then you've got this interesting class of organisms that are called facultative anaerobes. Facultative anaerobes have the ability to switch between fermentation or cellular respiration. If you're a facultative anaerobe, that's what we've got drawn here on the slide, you get some food and immediately you'll start doing glycolysis because both processes always begin with glycolysis. Once you get to the end of glycolysis, you then look and see what's available. If no oxygen is present, fine, you can go ahead and do fermentation, you won't die. If, however, oxygen is present, great. You have the ability to do aerobic cellular respiration. You'll get more ATP from that glucose molecule. We want to end by coming back to that weird enzyme I introduced when we started talking about glycolysis back at the very, very beginning of this lecture. This process of generating ATP has to be regulated. Cells have to make sure that they're using their resources wisely. So a cell is not going to waste all of its resources to make ATP if it already has 50 billion pieces of ATP. There needs to be some feedback mechanism. So we're going to use feedback inhibition, similar to what we saw in previous chapters. We're just revisiting it. In feedback inhibition, the end product will feed back and inhibit the enzyme in a pathway. I'm really only interested in one arrow on this particular diagram, so let's try to make sure we understand what's going on. If we look just moving up and down, this is trying to show us the process of cellular respiration. So here's glycolysis, there's the second step, pyruvate oxidation, citric acid cycle, and down at the bottom is oxidative phosphorylation. So if you go through this process straight down, you are making ATP. Again, if a cell has a lot of ATP, it's got 50 billion pieces of ATP floating around, we want to inhibit this process because we don't need to waste our resources making any more ATP. And so what ATP will do is ATP will feed back and it will inhibit the enzyme phosphofructokinase. This is the reason that you need to know the name of that enzyme. It's a very important regulatory molecule. It's how feedback inhibition works in aerobic cellular respiration. You do not need to know what's going on with AMP or citrate. I'm really only interested in how ATP can feed back and inhibit the enzyme phosphofructokinase. That gets us to the end of Chapter 9. If you have any questions about Chapter 9, either your reading or your materials, I encourage you always to reach out to me during online office hours. I would love to help provide clarity in anything that I may have spoken through too quickly or that you may have questions about.